Okay. Vitamins. Yeah, we're going to talk about vitamins per se. Now, I'm going to be very, very clear of what I expect you to know about vitamins and associated pathologies. So for fat-soluble vitamins, I do want you to know which vitamins are fat-soluble, which vitamins are water-soluble. Am I clear? So if I ask you, which of the following vitamins is water-soluble, you pick the right answer. Like K, A, B, or C, or more than one, and stuff like that. Vitamin A. Honestly, critical for vision. Probably most important. The low levels of vitamin A are associated with a condition called nyctalopia. Ever heard about it? Night blindness. When people cannot see clearly in the night vision is impaired, rods cannot lose sensitivity. For some reason, in my original tongue, if you translate it directly from Russian, it's called chicken blindness. I don't know why chicken, but chicken blindness. Vitamin A is really toxic. I mean, if you overdose on it. There was a story when a woman worked at the plant which was making vitamins. And vitamin A, since it's fat soluble, it is stored as the solution. And solution, it was back home, solution in sunflower oil. And she wasn't like involved in synthesis. And she didn't know what was that, and she thought, oh, we have like bottles of sunflower oil. If I will grab one and take it home, nobody will notice. And she took one bottle of vitamin A solution and brought it home and cooked the dinner for the family. And everybody died pretty much because it was like a million times more than lethal dose. Just highly concentrated solution just for storage. So vitamin A can be toxic. With the diet, you cannot possibly OD on vitamin A. You cannot eat that much carrot. Okay? Carrots, any colored vegetables, they contain a lot of vitamin A. For the best absorption of vitamin A, it should be consumed with a fat. When I was a kid, it was carrots with sour cream, always. Sour cream is fatty, it increases the vitamin A absorption. Sour cream, yeah. It increases absorption of vitamin A. Vitamin D, produced in skin. I mean, you can get it from dairy, okay? But it's produced in the skin, and it is necessary for calcium absorption. Again, if you would, I don't remember how I Googled it. It just popped up in my Google search. But if you think Ohio winter is long, think Russia or Canada. At some point, since sunlight is not readily available, and even if it is available, you wear a lot of clothing. Especially in the child care centers. It was back in communist time. It was all government-run child care centers, pretty much preschools. Kids like 4 or 5 years old, to avoid rickets, kids were exposed to UV light. And it looked incredibly cool. There was a bunch of kids in their undies with the shades standing in a circle around the UV lamp. It was like, I saw the picture. It looks like they're about to summon the Satan. Okay? It looked awesome. I don't remember if I was subjected to this. I was just probably too little. Vitamin E, antioxidant. Okay? I'm going to tell you what to know about these guys. Again, you can hardly OD on it because it turns out you cannot possibly eat that much food. Of course, if you will decide to take the entire bottle of vitamin A that you can buy in any pharmacy, then probably, yeah, it can be toxic. Vitamin K, critical for the blood clotting, and vitamin K is produced in sufficient amounts by the bacterial colon, really. Okay? Pathologies that I want you to know. Vision problems for vitamin A. Calcium retention, rickets. Remember, we talked about calcium absorption when we talk about endocrine. So, rickets for vitamin D, and problems with the clotting for vitamin K. Am I clear? These three? No. Just FYI, there were multiple studies suggesting that actually increased intake of vitamin D, like in the form of pills, reduces the risk for developing diabetes. Water-soluble vitamins. Vitamin B1. Its deficiency is associated with a disease called beriberi. The story of the beriberi discovery is fascinating. In fact, B1 was the first vitamin, beriberi, the disease, was observed in the Southeast Asia at the end of 19th century, beginning of 20th. Nobody knew what's going on until, I don't remember the names, as you know. I never remember, I'm terrible with names, but he got the Nobel Prize for it, not surprisingly. So that guy found a striking difference, that in areas where beriberi was common, people ate processed rice. When rice was collected, they husked it, like white rice. Where beriberi was not present, was absolutely no beriberi, people ate sort of a dirty rice. Rice with it's not shell, like a husk. That makes sense. So he figured that's probably what it is, and he managed to isolate the vitamin and showed that this vitamin really prevents beriberi, and then identified as thiamine, vitamin B1. So, neurological disorder, pretty much. Okay? Vitamin B2, and also can be found in grains. You can sometimes see the signs of vitamin B2 deficiency, mild deficiency, cracks in the corners of the mouth. When I was a kid, when I had this, the treatment, any physician would tell me, go and eat some rye bread. Rye bread is usually like pumpernickel bread. It is made from the whole grains, a lot of vitamin B2, cracks go away. B3, gastrointestinal lesions. Now B6, pyridoxine, anemia, muscle twitching, not really. Okay. B5. What I want to highlight about this vitamins B, if you would look at where you're going to find it, you will see that it's nuts, vegetables, grains, meat. Essentially, if you have a good diet with a variety of foods, you're going to get all those vitamins just from food. Does that make sense? Folic acid, necessary for amino acid synthesis. and nucleic acid synthesis. Folic acid is a critical vitamin for cell division. That's why pregnant women are often given folic acid as a supplement. Not because they're deficient, but to avoid the potential deficit. Does that make sense? Because it may not have pathological effects on the mother, but it might very well have pathological effect on the fetus. Does that make sense? Vitamin B12. Another vitamin that is critical for quickly dividing cells. Okay? Leads to anemia, which makes perfect sense because red blood cells, quickly dividing cells, often dividing, not red blood cells, but cells that precede red blood cells, precede erythrocytes, they divide all the time. And in the absence of vitamin B12 or at lower levels of vitamin B12, bone marrow will not be able to produce sufficient amount of red blood cells. That makes sense. Biotin, skin problems, vitamin C. That's a famous one, scurvy. Okay. Skin lesions, gum problems, bleeding gums, teeth fall out. Okay. And people die of scurvy. Now, before we go to the what I want you to know, which pathologist, vitamin C is still believed to prevent cold, be it treatment for cold or influenza. It's not. Multiple clinical studies show that there is no association between, recovery and drinking vitamin C. That's it. More importantly, daily requirements in vitamin C consumption, as far as I remember, is 300 milligrams. So if you buy like 1,000 milligram thing and you drink 1,000 milligram or 2,000 or 3,000 milligrams of vitamin C, that extra amount, you will pee it out. It's that. It's not going to stay in your body. It's not going to accumulate. Okay? The funny story about vitamin C, who started this whole hype about vitamin C and its protective effects on the common cold, was American scientist, chemist, Nobel Prize winner, Linus Pauling. He won first Nobel Prize in chemistry and second Nobel Prize, there was Peace Prize. So he was a strong believer that vitamin C prevents common cold. He was, as a Nobel Prize winner, he was invited to multiple conferences and blah, blah, blah. So he was invited to one medical conference and he gave a talk and pretty much he plugged in for vitamin C. He said, vitamin C is good, it's antioxidant. A lot of pathology that associated with common cold is because of inflammation. In inflammation, there's a lot of free radicals produced. Drink vitamin C, it will neutralize free radicals. And after he finished, a bunch of doctors who were present in the audience pretty much stood up and very politely said, then, yeah, it's all bullshit. Sorry, you're a chemist. We respect your Nobel Prize, blah, 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 but medicine says otherwise. So it was debunked right there. When the session was over, all these doctors who were present in the audience rushed to the closest pharmacy and bought all supplies of vitamin C. Because it's not going to hurt, and who knows, maybe he's right. So, but since then, many studies showed no, it doesn't really help. And you're going to pee it out. What I want you to know, the pathologies. This one, beriberi, folic acid, the birth defects, and anemia. B12, anemia, and vitamin C, scurvy. These ones, I can ask. Others, I won't. And God forbid, I'm not going to ask you to memorize which food is rich in what vitamin. It's not a nutrition class. And for this, honestly, there's Google. Seriously, there's Google. I do not like memorization for the sake of memorization. If you're not going to go into the dietetics, you're not going to remember that. Like if you're going to be a nurse in the, I don't know, ICU, I don't think that patients in ICU really care about broccoli. They have some other concerns. Minerals. There are macro elements and micro elements. There's a macro. Pretty obvious. So, calcium. You're going to get it from diet, mostly dairy. Okay. Phosphorus. What do you need phosphorus for? Which biological molecule? There's a lot of phosphorus in it. Say again? Phospholipids, yes. What else? Huh? Nucleotides. Yeah, DNA, RNA. Okay, so you need phosphorus. Sulfur. This is key. This is required for the protein synthesis, and it's going to be found in proteins. That's pretty much your major source of sulfur, proteins. Potassium, chlorine, sodium. Really salt. Potassium can be found, a lot of potassium can be found in the sea salt. Okay? That make sense? If you consume just the regular table salt, it's going to be sodium chloride, but you'll get potassium from other sources, from vegetables to meats. You cannot really go low on potassium, especially considering the fact that potassium is very tightly regulated, as we will see. We've already talked about aldosterone, right? Aldosterone regulates potassium in the blood very tight. And magnesium, which is an important cofactor. Again, you're going to get it from the food in sufficient amounts. Now, trace amounts. Iron. Well, if you run low on iron, you're going to develop iron deficient anemia. I would call it a good form of anemia, right? Because how you treat it, you just give someone iron. Not, you don't give them a dumbbell so they can, gnaw on it, but iron pills, there are drops with iron. Fluorine is necessary. For what? Fluorine. Teeth, yes. So fluorinated toothpaste. In many American cities, water is fluorinated to ensure that people consume enough fluorine with drinking water. Zinc is necessary for, it's element of DNA binding proteins. It's pretty much a cofactor for enzymes. You're going to get it. Same goes for copper. You're going to get it from the food. Basically, all your microelements, except maybe for fluorine, you can get from food. Okay. Associated pathologies. Know about iron, know about fluorine, and of course, know about iodine, which is critical for what? Thyroid hormone. Okay. Other than that, what I want you to be able to distinguish is to distinguish macro elements and micro elements. Does that make sense? So if I ask you, chromium is, you tell me it's a micro element. Does that make sense? Now, if you would look at this, it's practically all enzyme cofactors, which means these ions, these elements, are necessary for the proper functioning of various enzymes and various cellular processes. Now, I already warned you about the hype associated with various dietary recommendations. About 10-15 years ago, there was a huge hype about selenium. It's the microelement that is found in certain proteins, and it's a part of amino acid called selenocysteine. And proteins where this element is found participate in antioxidant activities in the cell. Does that make sense? They neutralize a lot of free radicals. So the idea was pretty straightforward. What if we, and there was some other evidence that deficiency in those proteins, if you like replace selenium with sulfur, you can do it. If you replace it, then it increases the risk for the cancer development and stuff like that. So the idea was, what if we start to give people selenium and see how it works against cancer? So there was a huge study called SELECT, carried out in the United States. It wasn't anything shady. It was completely legal. Huge study. Thousands of people were involved. And, they pretty much received placebo versus selenium supplements. The study was terminated early. Turned out that the group that was receiving selenium supplements overall demonstrated about 15% increase in cancer cases compared to the placebo group. Did selenium cause it? We don't know. I told you before, if you take two large groups and look at many parameters, you may see significant difference. They saw significant difference. They terminated the study. It definitely did not decrease the number of cancer cases. Does that make sense? So that kind of another illustration, take it with a grain of salt, all those dietary recommendations. Okay? Now I'm going to reiterate in terms of the elements: iron, fluorine, iodine, and difference between macro versus micro elements. Good? Now, metabolism. Metabolism, as the term, relates to all chemical reactions in the human body. Any reaction can be categorized as either catabolic or anabolic. Does that make sense? Catabolic is the breakdown. Anabolic is the synthesis. Can you give me an example of either catabolic or anabolic reaction? Anything. Example of catabolic reaction. Huh? Yeah, protein breakdown. Sure. Absolutely. Protein breakdown. Anabolic. Synthesis of proteins. Yep, that's anabolic reaction. Synthesis of nucleic acid, like DNA. DNA replication, pretty much synthesis of new DNA, is anabolic reaction. Breakdown of ATP is catabolic. Does that make sense? So this picture shows you catabolic and anabolic reactions in terms of the processing of nutrients. All the blue lines refer to catabolism. The purple lines refer to anabolism. So basically, the function of catabolic reactions generally release energy. If you break down ATP, energy is released. If you break down glucose, energy is released. If you break down lipids, energy is released. Does that make sense? Anabolic reactions, in general, use energy for synthesis. If you want to make ATP back, you need to take ADP and phosphate, okay, and use energy to put them together. If you want to make glycogen out of the glucose, you got to use ATP as the source of energy. That makes sense? Reactions that require energy are called endergonic. Endo, inside, ergo, energy. Reactions that produce energy are called exergonic. Does that make sense? So this image here shows you the reaction, the blue dial, wheel, whatever you call it, gear, the blue gear shows you the exergonic reaction. And the size of the gear is proportional to the amount of energy that it produces. So you can see that exergonic reactions produce more energy that endergonic use. That makes sense? So there's a difference. It is never 100% efficiency. Does that make sense what I'm saying? So you're never going to have a situation when 100% of energy from exergonic reaction will be used in the endergonic reaction. Some of that energy will be always lost in the form of what? No. ATP, you need to make ATP. Say, you consume food that gives you, I don't know, 2,000 calories worth of energy, okay? Chemically, it is worth of 2,000 calories of energy. Are you going to produce 2,000, are you going to produce ATP that is worth 2,000 calories? No, you're going to produce less. Where does the difference go? Yeah, that's why you're warm-blooded. Heat. Does that make sense? You spend about 60% of what you eat on the maintenance of the body temperature. If you think about it, we eat an enormous amount of food. Snakes, what do they need? Like once every 2 weeks? You give them a rat and that's fine. We have an enormous snake down the hallway. And she's 6 feet long, 20 pounds. You feed it once every 2 weeks. I have a dog who's 20 pounds. This animal eats twice a day, and when he has a chance, he jumps on the dining table, and if we can't see it, and just eats whatever's there. He's always hungry. He's warm-blooded. Snake is cold-blooded. The majority of the energy that we consume is used for heat. Does that make sense? Essentially, the energy that's released in a catabolic reactions is used in the process of glycolysis, Krebs cycle, oxidative phosphorylation, for the production of ATP. You have to understand that ATP is not the ultimate goal, because ATP is just the universal currency that human cells use, the energy currency. Does that make sense? So ATP synthesis is, of course, anabolic and endergonic. It uses energy. But ATP breakdown is exergonic. Does that make sense? You gotta know the difference between catabolism, anabolism, endergonic, exergonic. So, we're going to talk about basic biochemistry now. Oxidation and reduction is going to be a pain in the butt. This is the molecule of glucose. These are oxygens. Can you burn glucose? Just burn. Can you? Why not? Really can't. There's some tricks. Sugar is not going to burn, it's going to melt. You need to cover it with ashes, then it's going to burn. But yeah, you can burn glucose. And the amount of energy that will be produced in burning will be basically the same as the amount of energy that will be produced in the reaction of cellular respiration. And that's the reaction that we're going to focus on now. Does that make sense? Essentially, in cellular respiration, glucose is oxidized to produce carbon dioxide and water. Does that make sense? We're good? Okay. So, what is oxidation or reduction or both? What are those? When we say something is oxidized, what does that mean? Huh? Say again? As a chemist inside of me is infuriated. That's the biology. That's the biology speaking. It's not about hydrogens. You're right. Hydrogens will be exchanged, but chemically, it's electrons. Okay? So, if you have A plus B, two different chemicals, when electron goes from A to B, that's A oxidized, and B is reduced. When the molecule loses electron, it is oxidized. When the molecule gains electron, it is reduced. Now, electron is negatively charged. In order to keep it neutral, we have to transfer also what? Hydrogens. That's the way to keep it neutral. And that's actually, that's the difference between a colleague of mine, Dr. Landis. When he teaches oxidation and reduction, he focuses on hydrogens. That's biology speaking, because in the cell, and it's true, nobody cares about electrons. That's hydrogens, acid, that everybody cares about. Well, in college, I was majoring in chemistry. If I would say a word about hydrogen, I would be expelled. It was all about electrons, movement of electrons. Okay? So from chemical standpoint, it's electrons. And essentially, the energy conversions in the cell are about electrons. This molecule, glucose, has sort of high energy electrons inside. So what you need to do, you need to take these high energy electrons, you see how they're sitting on top of the stairs, and transfer them to potentially lower energy level in the molecule of carbon dioxide. When they transfer from top to bottom, they lose energy. That energy, essentially, this energy, is converted to ATP. Does that make sense? Good. So, two basic metabolic processes that happen in human cells are oxidative respiration, which reaction is shown here, okay, and lactic fermentation. When cells do not have oxygen, then glucose is not oxidized to carbon dioxide and water, it is oxidized to lactic acid. Does that make sense? Good. Now, a few important things. Since I'm the chemist, initially, we're going to be talking about electrons. Electrons cannot float around by themselves. They have to be carried. These are electron carriers, NAD and FAD. Okay? So when NAD receives electron, actually it receives 2 electrons. Okay, NAD receives electron, it also receives hydrogens. Does that make sense? What should we call this process? When NAD receives electrons, NAD molecule is reduced. When it loses electrons, and it will, it is oxidized. Now, the confusing part is that molecule that is reduced is the oxidant. Does that make sense? Okay. That's kind of a confusing part. And vice versa. Molecule that is oxidized is a reductant. Okay. Another electron carrier is FAD. FAD receives electrons and hydrogens becomes FADH2. So FADH2 is reduced form. FADH2 and NADH are reduced forms. FAD and NAD are oxidized forms of the same molecule. Does that make sense? This is the overview of the glucose metabolism. We've got glucose, starts with the glucose. In the process of glycolysis, glucose is broken down and partially oxidized to pyruvic acid. I'm going to overview and then we're going to talk about each step. That happens in the cytoplasm. Okay? Then pyruvic acid enters the matrix of mitochondria. We have Krebs cycle. Pyruvate is completely oxidized during Krebs cycle in mitochondria. And what you end up with is a bunch of electrons which are attached to NADH and FADH2. And these electrons enter the electron transport chain where the energy is used for the ATP synthesis. So glycolysis happens in the cytoplasm, Krebs cycle and electron transport chain are in the mitochondria. That makes sense? We're good? We're not going to be talking about 11 steps of glycolysis and enzymes that facilitate each of the steps. Actually, I personally think, I was asked to tutor a kid who takes AP biology in high school. And the first topic that they cover is cellular respiration. And they do have to know all 11 steps of glycolysis. I mean, why? No, seriously, because comprehending the importance of each step requires knowledge of thermodynamics, I'd be honest, I will not be able to tell you right now what is the rate limiting step of glycolysis. I don't remember. I can think, I can try to figure it out, but not right off the bat. In terms of biological importance, what are the key steps, key intermediates? First of all, in the cellular respiration, you have to count carbons. Does that make sense? So you have to remember that glucose has 6 carbons. We're good? You have a bunch of reactions, blah, 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 blah. You end up with these intermediates. 3 carbon intermediates that are phosphorylated. They constantly convert into each other. Does that make sense? Just chemically. So we're going to be using one of them as the poster child, G3P, or glyceraldehyde 3-phosphate. Okay? So 3 carbon intermediate. And this 3 carbon intermediate, well, 2 of them actually, become 2 molecules of pyruvic acid in the end. So far, can you follow me? We good? How many carbons did we start with? 6. How many carbons did we finish with? No, not 3. 6, because 2 molecules of pyruvic acid. Does that make sense? We didn't lose any carbons. Okay? What we produced, we did oxidize a little bit of glucose. Now, in order for the whole thing to run, we had to invest ATP in the beginning. So we had to invest 2 molecules of ATP at the first step, well, one of the first steps, and then we gained 4 molecules of ATP at the end. What's the net outcome? 2. Are we clear about it? 2 molecules of ATP. Is that a lot? That is not. What is more important, we produced NADH. What does it carry? Electrons. We got a first portion of high energy electrons. Make sense? You good? Awesome. We also produced 2 molecules of pyruvic acid, which is, I would say, more amenable substrate for energy conversions. What if you don't have oxygen? What's going to happen to pyruvate? It's going to become what? Lactic acid. Here's the tricky question. In anaerobic metabolism, in fermentation, what accepts electrons in the end? Which molecule is produced in the end? It's not an easy question. I didn't tell you this. You can get it from this flow chart. I will tell you where to look. What is that? What is the final electron acceptor in a fermentation? NAD carries it. Look at this. NAD, huh? No, it's anaerobic. We're talking about anaerobic now. Look, NAD here, can you see NAD? Accepts electrons and becomes NADH. Okay? And we get a pyruvate molecule. Then, NADH has to give electrons to something. If that would be respiration, it would be oxygen at the end. But now we want to figure out no oxygen. NADH, what it gives electrons to? What it gives electrons to? Huh? one step before. Not lactic acid, pyruvic acid. So pyruvate becomes the final electron acceptor in the fermentation. Does that make sense? And that gives you lactic acid in the end. That happens when there is no oxygen. That makes sense? So actually, if you look at the fermentation, and it happens mostly in the muscle, of course, if you look at the fermentation, it's not really energy effective. That makes sense. And it also produces lactic acid. What's the advantage of fermentation? There are two. One, it doesn't require oxygen, and number two, it's fast. It's fast. It gives you 2 ATPs only, but it's fast. It's important in muscles, especially, anaerobic effort, like fast glycolytic fibers. Does that make sense? Now that's about fermentation. Okay. Now what is when oxygen is present? What happens next? That's pyruvate entering Krebs cycle. You can think about it this way. Krebs cycle doors are really narrow, so 3-carbon molecule cannot squeeze in. Does that make sense? Since it cannot squeeze in, we get a trim one of the carbons of the pyruvate. This happens during the transition step or transition phase when pyruvate is converted to the molecule of acetyl-CoA. Now, in acetyl-CoA, you have coenzyme A, the blue thing, the blue circle, and is bound to 2-carbon, the acetyl residue. So we went through from 3 carbons to 2 carbons. That carbon that we lost, where did it go? CO2. So we produced a molecule of CO2. Does that make sense? Now, since carbon has been oxidized, the electrons that were ripped off the carbon, where did they go? NADH. So we produced another NADH molecule. Does that make sense? Then you have 2-carbon containing acetyl-CoA that interacts with 4-carbon containing oxaloacetic acid. 2 plus 4 gives you 6, which is citric acid. And that's the moment from which officially Krebs cycle starts. Okay? So 6-carbon citric acid is oxidized to ketoglutaric acid, 5 carbons. Where the extra carbon goes? CO2, molecule number 2. Right? Electrons allow us to form another NADH. Alpha-ketoglutaric acid is oxidized from 5-carbon to 4-carbon succinyl-CoA. Where does the extra carbon go? CO2. And the electrons are used to form NADH. Then you have a step of so-called substrate level phosphorylation. When succinyl-CoA is converted to succinic acid, produces 1 molecule of ATP. And then succinic acid, it's 4-carbon intermediate, through the series of steps, is converted to oxaloacetic acid. You don't lose any carbons. What you lose is hydrogens. Does that make sense? You oxidize. It's still oxidation, so with every round, through these 2 rounds of oxidations, you form 1 more NADH and 1 more FADH2. Okay? Now, let me summarize Krebs cycle We're going to do it together, and I will tell you which key steps you have to understand. So, you have one molecule of pyruvate entering transition phase. What happens to it first? Carbon, again, loses carbon. Carbon is oxidized. Electrons go to NADH, number 1. And you have CO2, number 1. Then it combines, 2-carbon acetyl-CoA combines with oxaloacetic acid, forms citric acid. Citric acid is oxidized to alpha-ketoglutaric acid. What is formed? CO2, NADH, number 2. Alpha-ketoglutaric acid is oxidized to succinyl-CoA, 4 carbons. CO2, NADH. 3 CO2s are the products, one of the products of the Krebs cycle. Forget about CO2s by now. We have 3 NADH molecules, right? You will produce one more NADH. And you will produce one FADH2. Does it make sense? Do you need to memorize things like citric acid, alpha-ketoglutaric acid? Not really. I want you to understand the number of carbons, really. So when you go from pyruvate to acetyl-CoA, you go from 3 to 2. When acetyl-CoA is combined with oxaloacetic acid, 2 plus 4. Does that make sense? How many Krebs cycles are completed per molecule of glucose? 2. 2 molecules of pyruvate, right? Does that make sense? So, if it's 2 Krebs cycles per 1 molecule of glucose, how many molecules of CO2 will be produced per molecule of glucose? 6. 6 molecules of CO2. How many carbons are in the molecule of glucose? 6. Krebs cycle completes the glucose oxidation. That its function. Complete glucose oxidation. Now, if we have 2 Krebs cycles per molecule of glucose, how many ATPs are going to be produced in Krebs cycle per molecule of glucose? 2. Is that a lot? It's not. So Krebs cycle is definitely not about ATP. Oh, sorry. The molecule of pyruvate, yeah. So this is your yield per molecule of pyruvate, right? I highlighted it there. Krebs cycle, what is it all about? It's about electrons. Basically what happens in the Krebs cycle, enzymes remove electrons from pyruvate and transfer them to NADH and FADH2. ATP is icing on the cake, really. The main thing is 4 NADH, okay, if we include transition step. 1, 2, 3, 4, and 1 molecule of FADH2 that are produced in the mitochondria. Does that make sense? So by this time, C6H12O6 plus O2, we produced CO2. Does that make sense? In terms of glucose oxidation, we produced CO2. Does that make sense? We did this. All we need to complete the reaction is to make water. Does that make sense? And this is where it is made. So as you remember, electron carriers, NADH and FADH2, along with electrons, they carry protons. Right? Remember, like a couple steps ago, we talked about the reduced and oxidized forms. So this is the reduced form, which carries electrons and protons. Does that make sense? This is the reduced form that carries electrons and protons. Is that clear? So these NADH and FADH2, these reduced forms in a mitochondria, they travel, they get to the proteins of electron transport chain. You don't have to memorize these proteins, but I do want you to know what they do. Essentially, proteins of electron transport chain, they take electrons, accept electrons from NADH and FADH2. So during this step, what happens to NADH and FADH2 in terms of oxidation or reduction? NADH and FADH2 they lose electrons, they are oxidized. Does that make sense? Proteins of electron transport chain are reduced, okay? And those electrons that are ripped from electron carriers, they go from protein to protein. That's why it is called chain. And at each step, they give energy away. Do I make sense? They lose, those electrons are high energy electrons, they lose energy. Proteins of electron transport chain, what do they use energy for? I didn't tell you this yet. Shown here. Huh? Not yet. Eventually, yes. But proteins of electron transport chain use energy of electrons to pump hydrogens across the mitochondrial membrane. Does that make sense? So you have a lot of hydrogens on one side of the membrane and much less on another. How do we call it? Huh? How do we call when there's a concentration difference? A gradient. Yeah, we have a gradient of hydrogen ions. Chemical gradient. What about charges? What's going to be the charge on this side of the membrane? Positive, and so we have not only chemical gradient, we also have electrical gradient. Altogether, it's called electrochemical gradient in the mitochondria. Does that make sense? So hydrogens, where do they want to go? Yeah, they want to go back to the negative side. Yeah, they want to go back. Can they? Not directly. Why? It's a diffusion. They should be able to follow the gradient without energy. What prevents them from going? The M word. The membrane, right? Membrane is hydrophobic inside, and they're charged. They can't cross the membrane unless there's a channel. So they all rush into that channel, and they practically fall. You know what I'm saying? They diffuse from high concentration to low concentration. They diffuse. And it's movement, it's kinetic energy involved. And that energy of diffusion is converted into the energy of ATP. So that's, Raquel, that's when the energy is used to attach phosphate to ADP. Does that make sense? Let me show you something. Oh, come on. So here's the deal. It's going to be you don't have to picture it. I'm not going to ask you questions like that. So think about this. Imagine that this is, imagine that this is the watermill, okay? I apologize for my artistic capabilities. Okay. So this is the pipe, and that's the water dripping on the watermill. Okay. Does that make sense? So this water dripping are hydrogen ions. And that watermill is ATP synthase. So you have kinetic energy of falling water that is converted into the energy of rotating watermill. So you have energy of hydrogen ions that is converted into the ATP. That makes sense? Now, see, it's a pipe. So it's not a river. So what brings water up there? There's a little pump right here, which is what? That pump is electron transport chain. Does that make sense? Now, what feeds that pump? There's a little dude that operates the pump. Well, let's say dude and the pump are proteins of electron transport chain. Now this little fella right here, he eats. He has a little lunchbox right here. So this little lunchbox are electrons from NADH and FADH2. Does that make sense? So this whole electron transport chain receives energy from electrons that come from NADH and FADH2. Does that make sense? Energy of these electrons is used to pump hydrogen ions across the mitochondrial membrane, which I don't picture here, and then it goes back strictly through the ATP synthase. Does that make sense? Strictly through ATP synthase. And that motion is what delivers the energy for the ATP synthesis. Does that make sense? So this whole thing, glycolysis, Krebs cycle, oxidative, oh, I'm sorry, I didn't notice. Like post-due, sorry, it requires a lot of time. So this is slow. What's the advantage? A lot of ATP. Basically, you can count it up. In ideal world, it's going to be 3 ATPs per NADH and 2 ATPs per FADH2. If that would be an ideal world, we would make 40 ATPs altogether, as far as I remember. Since the world is not ideal, with glycolysis and Krebs cycle and everything, it comes down to about 36. Okay? We're going to continue this on Monday. There's no quiz today. And we're going to talk about diet.